Let's begin with a review of glycolysis. There are 11 questions for you to try on Socrative to see how you're doing with this process. And I can't emphasize to you enough, don't begin this video unless you feel pretty confident about the process of metabolism or your understanding of metabolism and the process of glycolysis first. So pause the video and try the next 11 questions, including fill in the blank, until you get to the question that says this is the end of the review. So after a molecule donates electrons or its electrons density becomes lessened, this is what we know as oxidized. Now, an oxidizing agent refers to a molecule that will accept electrons in a redox reaction because it's going to be forcing another molecule to donate them. So oxidizing agents become reduced by forcing the oxidation of something else. Now, which of the following elements would be the best oxidizing agent? It would be the most electronegative, which is oxygen for the options that I've given you. Glycolysis occurs in the cytoplasm of the cell, and this video is going to switch over to talking about things which are happening in the mitochondria. How much net ATP is produced for a molecule of glucose in glycolysis? Recall that although we produced 4 ATP, we actually used 2 of them in that first energy investment phase. And how many pyruvate are produced for each molecule of glucose? 2. Glucose is a 6-carbon molecule, and so eventually, when we get to the end, we have to keep those 6 carbons. So how do you handle fill in the blanks like this? Well, here's a couple of things you can look for. Enzyme names can be clues. You'll notice that here the enzyme is hexokinase. Kinases transfer phosphates, so I'm already thinking about ATP. You can also count carbons. Well, if I were to take a look at my molecule of glucose here, I would notice that I have 1 carbon here, 1 carbon here, 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 and here. Each one of these carbons, or corners, is a carbon. And so we have 6 different carbons. If I look at my product, I have 1, 2, 3, 4, 5, 6. So I still have 6 carbons. The next thing I want to do is count phosphates. I see no phosphates on my first molecule. But this is a kinase, so I'm paying attention to the fact that now I have a phosphate in my second molecule. And so something has to happen, has to occur, that's going to add a phosphate here. Well, phosphates can't appear or disappear. And I know that I can go from a molecule of ATP to a molecule of ADP, and this will allow me to give this phosphate here. It's very important to pay attention to the directionality of this arrow and the directionality of the fact that since we're gaining a phosphate on our second molecule here, that we're going from ATP to ADP in this example. So what enzyme catalyzes the step shown here? Well, we still have ATP and ADP. So that already tells me that I'm suspecting my enzyme name is going to include the word kinase. In glycolysis, which has already been identified for you, there's only one name of a kinase that you need to know, and that's phosphofructokinase. What about this molecule here? Well, I know that I have nothing here that's going to change the number of carbons, so I need to keep them the same. I have 1, 2, 3, 4, 5, 6 carbons. So my final molecule is still going to have 6 carbons. I also have 1 phosphate, but since I'm going from ATP to ADP, I know that I'm going to be going from 1 phosphate to 2 phosphates. And indeed, that's the molecule you see here, where we've added a phosphate onto this particular carbon here. which is just symmetrical of what we had done before. Now, how would you name this particular phase? Would you say that hydrolyzing ATP would be an investment of energy or a payoff? This is an investment because you've hydrolyzed ATP. Now, in this particular model, we're going from a 3-carbon, 1-phosphate to a 3-carbon, 2-phosphate. So your first instinct might be to simply place an ATP to an ADP here. But we know that this is the payoff phase. We have a dehydrogenase and 2 phosphates which are being added into the system. So if this is the source of my phosphates, that means that I'm not using ATP. And dehydrogenases don't hydrolyze a molecule of ATP. They reduce a molecule of NAD+ to a molecule of NADH. And in fact, that's exactly what we see here. Once the cell has gone through and broken down this molecule of glucose into these smaller pyruvate molecules, the next step is going to be oxidize those pyruvate molecules. The oxidation of pyruvate has nothing to do with oxygen. The oxidation is referring to a redox reaction, whereby we're oxidizing this molecule and reducing a molecule of NAD+. Well, the enzyme that reduces NAD+ to NADH is dehydrogenase. And it turns out that this process actually occurs in a multi-step function. Pyruvate from the cytoplasm, where we did this process of glycolysis, has a molecule of carbon which is removed. This carbon comes off as CO2. This is going to be always the form in which this carbon is removed. At the same time, the hydrogens released from this process are used to reduce a molecule of NAD+ to a molecule of NADH. And then that bond is replaced using a molecule called coenzyme A. And so we named this final molecule, the result of the oxidization of pyruvate, acetyl-CoA. We've moved now from this glycolysis in the cytoplasm into the mitochondria. So tell me in Socrative, what molecule would you place in the blank here? This molecule up at the top is pyruvate. And so each one of these bubbles represents one of the carbons in this 3-carbon molecule of pyruvate. Here, acetyl-CoA has just 2 carbons. So we are going from a 3-carbon molecule down to a 2-carbon molecule. A couple of things have to happen. If we lose a carbon, we also need to make sure we can account for those electrons and reduce a molecule to pair with it. Well, here's NAD+ to NADH. That's going to be our reduction molecule. Here's CoA, which is the enzyme that was attached. We can see that. So we need to somehow account for the carbon that was lost, going from 3 to 2. It turns out when you lose a carbon, this is actually CO2. And so that's going to be our missing component. When you take a look at how this acetyl-CoA is used, this is a cycle. And we're going to work through each one of these steps of the cycle. But ultimately, the products of the cycle are very straightforward. For each time that we lose a molecule of carbon dioxide, we have to reduce a molecule of NADH. But this process of extracting the carbons leaves us needing to recreate the original molecules we started with, because this is a cycle. And so we're going to systematically break down this product by extracting an ATP, FADH2, and a final molecule of NADH before we can actually begin the process again. So there is a little bit of ATP which is going to be produced here by substrate level phosphorylation. Now the citric acid cycle has 8 steps. Again, if you plan on taking a graduate school exam, you can expect that they're going to want you to know what each of these molecules are, the enzymes that catalyze this production, and exactly how the cycle works. But for this course, we just want you to get a good idea of the nature of this process and be able to do those fill-in-the-blank type questions like you did with glycolysis. You need to start by being able to name molecules that are in the cycle so you can describe them. You already know acetyl-CoA because acetyl-CoA is the product of the oxidation of pyruvate. And acetyl-CoA is going to join with the 4-carbon molecule known as oxaloacetate. Since acetyl-CoA has 2 carbons and oxaloacetate has 4, the final product which we make is citrate, a 6-carbon molecule. And the name citric acid cycle comes from the fact we begin this process by creating citrate. The next 7 steps decompose citrate back to oxaloacetate. And this is what actually creates this process as a cycle, because we take a citrate, go back to oxaloacetate, and then can combine with a new acetyl-CoA, which creates a new citrate, and repeat the cycle over and over again. In the end, that 7 reaction process to recreate oxaloacetate yields 3 molecules of NADH, 1 FADH2, as well as a molecule of ATP and 2 carbon dioxide per cycle. So you'll actually double this for each glucose. Make note of the fact that this is a cycle. And if you're asked to describe why this is a cycle, you need to actually do the same thing. A cycle is something that starts and ends at the same place. So your answer needs to start and end at the same place. If you start by describing acetyl-CoA and its attachment to oxaloacetate, make sure that your answer includes that these create citrate, which decomposes back to oxaloacetate and can recombine with a new acetyl-CoA. My answer started and ended at the same place. So here's the addition of acetyl-CoA to oxaloacetate. This forms the 6-carbon citrate. That 6-carbon citrate is then going to be oxidized, and so a molecule of NADH is reduced and a CO2 is lost. That takes us from a 6-carbon molecule into a 5-carbon molecule. This 5-carbon molecule, the same thing happens. A CO2 is lost, an NADH is reduced, and now we're left with a 4-carbon molecule. This 4-carbon molecule here at the end ends with our CoA group being reattached. And so that CoA group allows us to be able to go in, utilize an inorganic phosphate, and actually create a molecule of ATP. Technically, we create a GTP, and then that GTP is going to be kind of like a placeholder for ATP. At some point later this semester, I'm going to show you how GTP is used by cells, but since this isn't something you understand yet, all I'm expecting for you to be able to do is to discuss how this stage produces ATP. Now, ATP's production is going to leave us with a still 4-carbon molecule, but that 4-carbon molecule is going to actually result in a reduction of FADH2, and then this little central double bond. Once we go through the process of moving that bond around, so we're back to single bonds, we can go through and reduce another molecule of NADH, creating this terminal double oxygen bond. And that's the difference between creating an FADH2 and an NADH. But you'll take note that now we are back to oxaloacetate, which was our starting molecule to begin with. If you take a look at this process, you should be able to count some carbons and see us going from 6 to 5 to 4, and how each one, oh, this one's still 6, I apologize, to 5 to 4, and how as we go through this process, you lose a carbon each time that that you go through this as a molecule of CO2. Each molecule of CO2 that's lost also gets paired with the reduction of an NADH. And then there are 3 remaining steps, one which makes ATP, one which makes FADH2, and one of which makes NADH, in that order. There are only 3 names that you have to know here for the citric acid cycle: oxaloacetate, acetyl-CoA, and citrate, but you'll need to be able to utilize those in order to describe the cycle. Finally, you should be paying attention to the fact that although 1 glucose feeds into glycolysis, 2 pyruvate come out as a product, which means that per glucose, the oxidation of pyruvate happens twice. And the citric acid cycle goes through 2 turns. So anytime that you're calculating yield per glucose, you need to be cognizant of the fact that some of these processes actually happen more than once.